Now we're going to spend some time talking about the most abundant protein in the human body. Of course, that's collagen. Not only is collagen the most abundant protein in the human body, but it's a protein that's frequently going to have problems with it that's going to lead to human disease. And as a matter of fact, there's a lot of different types of collagen. You don't have to study and remember every single type of collagen, but there's a subset of collagen types that you will have to know that you will be tested on. So let's look at a table. Now, why do we pay attention to the different types of collagen? It's because there's disease that are associated with defects in the different types of collagen, and you're expected to know which diseases are caused by a defect in which type of collagen. So let's look. Now, collagen type 1. A defect in collagen type 1 is the most common cause of osteogenesis imperfecta, and we'll see that in a minute here. But osteogenesis imperfecta could be caused by a defect in collagen 1 synthesis or a mutation to the collagen 1 coding sequence. Also, various types of Ehlers-Danlos syndrome can be caused by a defect in collagen 1. Collagen 2, not very highly associated with any specific disease. If you're presented with a patient that has defects in cartilage or maybe vitreous humor of their eye formation and you're asked which type of collagen is it, just remember it's type 2 collagen. Type 2 collagen is cartilage and also vitreous humor. Collagen type 3. There's Ehlers-Danlos type 4, which is a common cause of Ehlers-Danlos syndrome, is caused by a defect in collagen type 3. Now, probably the most important thing for you to remember is that patients that are predisposed towards keloids, so typically African Americans would be these types of patients, keloid formation is actually granulation tissue, which is made up of collagen type 3. So in a patient that's predisposed to keloids after they get cut or injured in some way, perhaps through piercing of the ear, patients sometimes get these large keloids. Those keloids are composed of collagen type 3 that was never replaced with the strong collagen type 1. Now, a board favorite is probably the collagen type 4. Collagen type 4 is associated with Goodpasture syndrome, with Alport's disease, with epidermolysis bullosa. In fact, in Goodpasture's disease, you see antibodies produced against your basement membranes. And it gives you this nice linear staining pattern on an immunofluorescence stain because the autoantibodies are made against the type 4 collagen of the basement membrane. So you get a nice autoimmune attack. It's going to cause eventually renal failure, renal problems in a patient with Goodpasture's disease. Also going to eventually perhaps lead to pulmonary problems. Now, Alport's disease is a defect in collagen 4 synthesis. It's also going to lead to a problem with kidney functioning. Epidermolysis bullosa is an autoimmune disease where you're going to get attack of type 4 collagen and you get blistering of the skin because you see the defect in the basement membranes. So the connective tissue isn't held together in a strong way. You'll get some of that reviewed again in pathology, but the biochemical importance of many of these are going to be discussed now. For collagen synthesis, in collagen synthesis, if you remember, collagen is simply a repeating tripeptide. It's a glycine and then 2 amino acids, a glycine and then 2 amino acids. So every third amino acid in the collagen molecule is glycine. Glycine is a very small functional R group. It's just a hydrogen atom. So this allows collagen to form that nice alpha helical structure or beta pleated sheet structure to give it some uniformity and some strength. Now, to synthesize collagen, it's initially synthesized on a ribosome, which through an N-hydrophobic signal sequence is then extruded into the endoplasmic reticulum. We call this pre-pro alpha collagen. So the hydrophobic signal sequence helps the pre-pro alpha chain of collagen get into the endoplasmic reticulum. Immediately, the signal's peptide is going to be cleaved off by the signal peptidase. So at this point, we now have a pro-alpha chain of collagen. Pro-alpha chain of collagen. The next step is a very important one for collagen synthesis, and it's hydroxylation of selected proline and lysine residues. So you now see hydroxyl groups being attached to the pro-alpha chain of collagen. This is a very important step. It requires vitamin C. Remember that, it requires vitamin C. The next step after hydroxylation is glycosylation. So actually the reason that the collagen had to get hydroxylated was so that later it could get sugars attached to some of those hydroxyl groups. Some of the hydroxyl groups get sugars attached, some are just left as hydroxyl groups. And the last step that happens with the endoplasmic reticulum is the association of the pro-alpha chains into a triple helix. So 3 pro-alpha chains can come together to form a triple helix of collagen. At this point, the collagen can then be secreted out into the cytoplasm. So it goes from the rough endoplasmic reticulum out into the cytoplasm. Just to remind you, the vitamin C dependent step here is shown with my cursor here. It's the proline and lysine hydroxylase. Proline and lysine hydroxylase require vitamin C. If vitamin C is deficient, what disease do we get? Scurvy, right? You get scurvy. You get poor wound healing, poor connective tissue, you get extra bleeding, you could get anemic, okay? It's the disease of pirates because pirates didn't have enough vitamin C on board their ships. So you became a scurvy little pirate. And then the British sailors got smart and they started carrying limes on the board the ship with them so they didn't get scurvy. So that's why they started calling the British sailors limeys, right? Okay, you knew all this. So not only scurvy, but Ehlers-Danlos syndrome. Ehlers-Danlos syndrome, most types of Ehlers-Danlos syndrome, the ones you'll be tested on, are caused by a defect in lysine hydroxylase. So the enzyme that adds these hydroxyl groups to lysine residues within the collagen protein, well, if there's a defect, that leads to Ehlers-Danlos. And then finally, osteogenesis imperfecta. Osteogenesis imperfecta is a defect either in the synthesis of collagen or the collagen that gets synthesized has mutations in it that cause it to be quite weak. So when the collagen goes to associate down here into triple helices, the association is very weak and the collagen crumbles and falls apart and gets brittle. So another name for osteogenesis imperfecta is brittle bone disease. Brittle bone disease. Now, after the collagen gets secreted out into the cytoplasm and then secreted from the cell, what's going to happen to it? There's further modification that happens. So once it's out in the extracellular matrix, the collagen propeptides are going to be further clipped. So these little edges of the triple helix are going to be cleaved off, and now we call this tropocollagen. Tropocollagen. The tropocollagen is going to later assemble into fibrils, to strong, tight, structural collagen fibrils. In order for this to happen, there's a special enzymatic reaction that has to happen, and that's shown on the slide. This is called lysyl oxidase. Lysyl oxidase comes along, it oxidizes the collagen, and allows the collagen to assemble into clumps or fibers. It's important for you to remember that lysyl oxidase is a copper requiring enzyme. Do you remember the name of the disease when there's a problem in getting copper into the proper place? Okay? This disease isn't a defect in lysyl oxidase. It's a defect in getting the copper to where it's needed for lysyl oxidase activity. Well, there's a couple of copper diseases, but the one that causes the most problem with collagen is Menkes disease. We'll review that in just a second. Menkes disease. Finally, once the lysyl oxidase activity binds to, reacts with the collagen, it allows these fibers to associate, the fibrils associate into a fiber. You have a nice, strong collagen fiber within your extracellular matrix. Oxidation of lysine, right? In order for this cross-linking to form, you need this lysine hydroxylase. So if ever there's a problem presented to you on the test where there's a problem with lysyl oxidase, also known as lysine oxidase, or a problem in getting copper where it needs to go, there's going to be defective collagen cross-linking. So now let's consider a very important high-yield table. These are disorders of collagen biosynthesis, some of which we've already talked about, but let's review them again from a clinical perspective. Scurvy. Scurvy is going to present with frequent bruising, little petechiae and ecchymoses. You see poor dentition, so you get loose teeth, bleeding gums, the wound healing is poor bone development, and something you might want to add in there, anemia. Anemia is a hallmark of scurvy, right? Because not only is vitamin C important for the collagen assembly, but vitamin C is also important for the oxidation reduction of iron. So if vitamin C is not there for oxidation reduction of iron, you're going to get poor iron association with red blood cells. So another hallmark of vitamin C is anemia, vitamin C deficiency. But as far as collagen goes, a vitamin C deficiency is going to cause deficient hydroxylation because vitamin C or ascorbic acid is missing. Moving on to osteogenesis imperfecta. In osteogenesis imperfecta, there's mutations in the collagen gene themselves. Usually it's collagen type 1. These mutations can happen to the coding region, so the collagen that gets made is weak, crumbly, poor collagen, or the mutations can be to the promoter region, so that you're getting good collagen made, but you're not getting enough of it made. So there might be mutations to some of your promoter elements that cause less collagen to be produced. Okay, we'll review those promoter elements a little bit more in a minute, but the promoter elements, remember, are things like the Tata box, the cat box, and then there's other enhancer regions that we'll speak about. An individual with osteogenesis imperfecta is going to have skeletal deformities, frequent fractures. They may or may not have blue sclera. This, although may seemingly pathognomonic for osteogenesis imperfecta, you might see blue sclera in some forms of Ehlers-Danlos. And for that matter, you don't see blue sclera in all forms of osteogenesis imperfecta. So just keep that in mind. Blue sclera doesn't automatically assume osteogenesis imperfecta, although you will see it sometimes. The skeletal deformities and fractures, you're always going to get a case of this where they're going to want to put in child abuse versus osteogenesis imperfecta on the differential diagnosis. So just remember to review the types of fractures that would typically happen here, fractures to long bones and so forth. Whereas you have, in child abuse, you have a certain pattern of fractures that might tend to occur. Ehlers-Danlos syndromes. There's various different types, as we'd said. It's caused by mutations, sometimes to the collagen genes, but the one you'd be tested about is a mutation to the lysine hydroxylase. Some mutations happen where the lysines themselves, which get hydroxylated, might be mutated, but most of the time it's the lysine hydroxylase enzyme that accumulates the mutation. I like to think of this as Ehlers-Danlos instead of Ehlers-Danlos. Why? Because the skin's loose. You can stretch it. You can bend it. Okay, so in Ehlers-Danlos syndrome, the skin's very loose. You get hyperextensible, hypermobile joints. The skin itself is very fragile. You may or may not get blue sclera. You get varicosities. You get ecchymoses because the blood vessels themselves are a little weak. And probably, other than somebody being tall and lanky and hypermobile and hyperextensible and able to do circus contortion acts, the real problem here is that these patients are prone to intestinal ruptures and arterial ruptures. Actually, Ehlers-Danlos syndrome is highly associated with a specific type of aneurysm. It happens up in the anterior communicating branch of the brain, in the circle of Willis. Do you remember that type of aneurysm? It's called a baryaneurysm, right? A baryaneurysm in the anterior communicating branch of the circle of Willis. This can rupture and lead to subarachnoid hemorrhage. Okay, so Ehlers-Danlos syndrome, polycystic kidney disease, those are two diseases highly associated with those baryaneurysms. You can also get aortic dissections with Ehlers-Danlos. Menkes disease. In Menkes disease, the problem is in the intestines. There's a protein that usually pumps copper that's absorbed in the intestinal epithelial cells. There's a problem in pumping the copper out of the intestinal epithelial cells into the portal circulation. So in Menkes disease, it's really a secondary copper deficiency. The primary problem is in a copper efflux protein. So getting the copper out of the intestinal epithelial cells is the problem. So if all the copper gets trapped inside the intestinal epithelial cells, it doesn't get dispersed throughout the body to go to the places it needs to. As a result, in Menkes disease, there'll be some copper that gets through, but not enough. So the patient's going to get depigmented steely hair. Why is that? Why would a patient with Menkes disease that has a secondary copper deficiency have depigmented hair? What does that have to do with collagen? Actually, it has nothing to do with collagen. It has to do with the fact that the enzyme that makes melanin, or helps synthesize melanin, converts tyrosine into melanin, the pigment. That's called tyrosinase. That's a copper-requiring enzyme. Okay, so the depigmented steely hair helps you recognize Menkes disease because copper is required for the melanin production, but it has nothing to do with collagen. The collagen problems are shown down here. So in a patient with Menkes disease, there's tortuous arteries. These are prone to rupture. So once again, you can get dissecting aortas, and you can get abdominal aortic aneurysms happening. You can even get cerebral degeneration. These patients are prone to osteoporosis and maybe even anemias. So in Menkes disease, you're going to see this copper problem. Its copper can't get delivered to other tissues. Don't confuse Menkes disease with another highly tested copper disease. And what is that? It's Wilson's disease, okay? Wilson's disease is another copper problem, but the problem in Wilson's disease is in the liver. So the copper gets from the intestines to the liver, but it gets trapped in the liver. As a matter of fact, the copper efflux protein that pumps the copper out of the liver is dysfunctional. So in Wilson's disease, you see perhaps problems with collagen, but it's more psychological problems. Patients with Wilson's disease, they're going to get a little paranoid, maybe a little confused, and unfortunately, they're going to get liver cirrhosis from all that copper accumulating within the liver. Now, the pathognomonic presentation of Wilson's disease, you guys all know this, is what? It's in the eyes, Kaiser-Fleischer rings in the eyes. They accumulate within Decimet's membrane up in the eye, and you can look in a patient's eye with an ophthalmoscope, and many times you can see these Kaiser-Fleischer rings. Another thing about Wilson's disease is that you see a decrease in ceruloplasmin. Ceruloplasmin is an important copper-carrying protein through the blood, and you would see decreased levels in Wilson's disease. So that's going to bring us to a close in Chapter 4. There was a lot of information here involving the genetic code, mutations that happen to cause problems with proteins, the mechanism of translation, and even post-translational modification. One of the most important proteins, the most abundant protein in your body, is collagen. So it's going to be very important for you to know specifically how collagen is post-translationally modified, because there's many different diseases you'll see in pathology where there was a defective collagen synthesis, or there was a problem in modification in collagen, and these are types of questions that you'll be tested on as far as the biochemistry goes.